All right, we're going to start with Unit 6, Gene and Protein Regulation. Our phrase for this unit is differential gene expression, and this is one of those phrases that I love and we say over and over again repeatedly throughout the unit. Next unit on evolution will also refer to differential gene expression in another context. And I have to be honest with you here, this unit is going to be introducing to you a lot of different concepts, stuff that you have not seen before in other courses. There is some detail that you have seen in other courses, but a lot of the stuff is going to be brand new to you. So do want to spend some extra time with this, looking at it, and you're going to learn things in a lot more in depth here than you learned in prior classes. So let's go ahead and get started. Our first essential knowledge idea is that gene regulation results in differential gene expression, and that leads to cell specialization. So we're going to talk first about differential gene expression, and we have two examples for you, very simple examples of prokaryotes, and those are the operons. And basically an operon is a group of genes that are transcribed together that code for a set of proteins. In the case of the lac operon here, we see lac Z, lac Y, and lac A. They're going to be coding for 3 different enzymes, and then those enzymes, their job is to carry out a metabolic pathway, which is to digest lactose. And lactose, you know, is the sugar that's found in milk. So you could give bacteria that contain the lac operon lactose as an energy source. The one we have over here is trip operon, and tryptophan is one of our amino acids. You may know it from its role in supposedly making you sleepy when you eat turkey at Thanksgiving. Doesn't turn out to quite be the case, but it's the same idea. But you see again, there are 5 different proteins that are going to be coded for, and they are going to be carrying out synthesis or building up of tryptophan amino acids. So lac operon is what's called an inducible operon, and what it does is it's got this repressor right here, this one protein. It's actually bound to the DNA, and it blocks RNA polymerase from transcribing the genes. Now it's turned off unless lactose is present. When lactose is present, it binds to the repressor and turns it off. That allows RNA polymerase to bind to the DNA sequence, code for the the proteins, and then have those proteins then go digest lactose. When the lactose is digested, the repressor goes back in and it's turned off. It's inducible. Basically, it's an on-off switch. And it's kind of the opposite thing here with tryptophan. Essentially, tryptophan is going to be, the repressor is going to be inactivated, and so that means that tryptophan synthesis is always on. And then the only reason it's turned off is because tryptophan is present. Tryptophan binds to the repressor, and then it binds to that complex binds to the DNA and keeps the RNA polymerase from binding to it. So when you have a lot of tryptophan, it's going to be turned off. When you don't have very much tryptophan, it's going to be turned on. So those are 2 very simple examples of what's called differential gene expression. We can turn genes on or we can turn them off. Now, we see the same kinds of things in our eukaryotic DNA, but it's a little more complex. Eukaryotic DNA cannot be directly transcribed and then translated. They have these other segments in there that are within a particular gene, which are non-coding segments, and probably not heard about these, so we'll introduce some of the ideas to you. First, the gene itself starts here in the blue and ends right down here at the terminator. And it actually has these 2 regions that are called exons and introns. And the exons are the coding regions, and the introns are going to be the non-coding regions. So after this is transcribed by RNA polymerase, the introns themselves will be removed, the exons will be reassembled. So the DNA has different non-coding sequences even within the gene itself. Outside of the gene, we have these control sequences. The first one is a promoter, and there's going to be this very fun phrase we'll talk about called the Tata box, T-A-T-A. And that is where the RNA polymerase is always going to bind to start any kind of transcription. And beyond the Tata box, a promoter, they're going to have these near control elements, and then they're going to have these far control elements. And these are segments of DNA, coding segments of DNA, that we have transcription factors, which are different proteins, that will bind to these, allowing the RNA polymerase to go ahead and bind to it and go ahead and get started with transcription. So DNA has coding segments and non-coding segments. Within the gene, remember that a gene codes for a protein. Not all of the segments within the DNA that code for the gene or code for the protein are actually going to be coding. There'll be some non-coding segments. And then we're going to have a promoter, and then we're going to have these proximal and distal control elements that will bind to proteins and allow the DNA to be transcribed. So there's a lot of stuff going on with DNA that you've not seen before, and so you want to kind of get familiar with these guys. All right, so then we also have a lot of communication that is going on both within the cell and outside the cell, which is going to affect gene expression. And a good example of that is a protein called the tumor suppressor gene, P53. You may possibly have heard of mutations in P53 that are associated with cancer. And it's true. That's one of the primary ways that we know that a cancer has taken place. If we look at the P53 gene in cancers, cells that are cancerous, we're going to find the P53 gene has been mutated. So let me give you a simple example right over here. We have UV radiation from sunlight, and one of the effects of that UV radiation is to cause damage to DNA. That is going to cause an intracellular chemical message to go to activate P53. The P53 is going to help repair the DNA, or it's going to cause the transcription of proteins that will block the cell cycle. Sometimes they can cause the cell to die, sometimes they can cause it to stop replicating, but in any case, they're not going to form a tumor. Right? So that's a pretty simple example. P53 is going to either help repair the DNA, or it is going to cause the cell itself to either die or to stop replicating. In either case, it won't form a tumor. So now, it doesn't just have to be UV radiation that can cause this. Damaging agents can be things like X-rays, free radicals, particular kinds of chemicals. We already talked about UV radiation, and even simple replication errors. And there's going to be basically three results. We may have a slowdown in the cell cycle. It may not replicate for a while. We can have the initiation of what's called apoptosis, or that's the programmed cell death. That's a control mechanism that all cells contain. Or we could get mutations which cause cancers. And those are going to be, or of course, we could pass those on to our children, depending on the type of cell it is. So, tumor suppressor genes, they're going to get these intracellular chemical signals that say, hey, we've got some damaged DNA, and they're going to respond to those. A mutation in those can cause cell death, or it can cause tumors to form. Now, you might think that it's just one mutation that would cause cancer, but it turns out that that's not the case at all. And if we look at the little example here, we have our little sports car. The tumor suppressor genes are keeping it from doing anything. It's not going to die. Nothing bad's going to happen to it. If we get one mutation, it's going to get a little bit closer towards cancer, but it's not really going to get there. Sometimes it requires a second or a third mutation. So we have the one mutation here, and then we have a deletion here. And now we get the tumor suppression process fails, and the cell becomes cancerous. Now, during your lifetime, you're going to have thousands and thousands of cancerous cells form, but very few of them will form tumors because they typically do it in an isolated fashion. And we have other control mechanisms that are going to keep them from becoming cancerous. So don't think because you go out today and get lots of sun that you're going to get cancer from it and die. That just doesn't happen. Right, another thing that we have are called Homeobox genes, and these guys are super cool. And these basically are a shared set of genes that are found in all animals. And it doesn't matter what kind of animal it is. I mean, if you look, we're pretty far away from a fruit fly, and yet we share lots of genes in common with the fruit fly. And many of the genes that we share in common have to do with the body plan. And you can see that the order of these genes based on color is going to determine location of different body parts. This little red part you see right here, you can see right up there. Now, what is different between the Drosophila and us has to do with the fact that we are going to have these gene duplications, where they may have one copy of the gene, we may have two, they may have one, we may have 4 duplications of it. And that is going to give us the differences in our body plan, but the genes themselves are the same. So if you're looking at this, you're like, okay, how could we possibly look like a fruit fly? Well, the reality is that the control mechanisms for the same gene expression is going to be the thing that matters, how the gene is expressed, where, and when. All right, so now this is something you have heard before. Changes in genotype result in changes in phenotype. And the phenotype, of course, is your physical characteristic. You learned that in Mendel earlier this year, and you also learned that, of course, in your other classes where you learned about Mendel. And we know that most of the changes in genotype are the result of genetic mutations. So Mutations, again, are changes in DNA sequences. They can happen at the gene level, they can happen at the chromosome level. I put up for you here 3 different mutations that are at the chromosome level. And this first one over here is called polyploidy. Pull that baby up, resize it for you. And polyploidy is going to be where two different plant species are going to have the pollen from one fertilize the egg of the other, and when the sperm fertilizes the egg in there, it produces a hybrid. And instead of having the same number of chromosomes, they may have twice as many chromosomes. You may find this hard to believe, but we have 23 pairs of chromosomes in humans, and corn, something that we eat, almost everything we have has corn in it, actually has 112 total chromosomes, 56 pairs, and that's a result of polyploidy. And polyploidy can cause something called hybrid vigor. So you see here that the hybrid between these two species is going to be growing much more rapidly, producing larger flowers than either one of the initial species did. So hybrid vigor is a positive kind of mutation as a result of polyploidy. We can't really do polyploidy in animals. Typically, when chromosome numbers are wrong, we're supposed to have 2 of each form of a chromosome, then we're going to get an organism that doesn't develop. On occasion, we get some that do, and 2 examples of those, you've probably heard of Down syndrome. And in Down syndrome, we have a situation where we have an extra chromosome, in this case, an extra chromosome 21. Down syndrome can result from other mutations, other chromosome mutations, but the most common one is carrying an extra chromosome 21. We also have another one called Turner syndrome, and you probably heard about this one as well. And in Turner syndrome, we only have 1 X chromosome, and we do not have a Y chromosome. So instead of having, or another X chromosome, so instead of having 46 chromosomes, we only have 45. So this one is called trisomy because we have 3, and this one over here is called monosomy, so we only have 1 in a pair. And reverse polyploidy is another one. But mutations are creating genetic variations. So some very simple ones that you already know, like deletions and duplications and insertions, and then some other more complex ones like we see here. So changes in genotype do result in changes in phenotype. And another example of that, differential gene expression and genotype, is what's called heterozygous advantage. And in this case right here, we have sickle cell anemia. And sickle cell anemia causes blood cells to have inappropriately shaped hemoglobin molecules. And those exposed to low oxygen cause the red blood cells to sickle, and hence the name. So they're not sickled all the time, they're not all sickled, but what you get is you get people who are in places in the world where we have exposure to malaria. The sickle cell apparently is not susceptible to getting infected by the malaria parasite. And so people who are normal, they're going to die if they get exposed to malaria. They have a higher rate of death. And those who have the sickle cell themselves are also going to die because the sickle cells are going to cause tissue and organ failure. And the people who are heterozygotes are the ones that are more likely to survive and reproduce because they're less likely to get infected by malaria, and they're less likely to be affected by the sickle cell itself. So if you look in places in the world where they have lots and lots of malaria, like the pink shaded over here, we're going to see in those areas high levels of the frequency of the sickle cell heterozygotes. And just like with hybrid vigor, these people are going to be more likely to survive and reproduce. When they have children, some of their children will be normal, some will have the sickle cell, but then about 50% of them will have they'll be heterozygotes. Another example of differential gene expression that it's going to cause, in this case, heterozygote advantage. All right, well, we also have many different ways in which we can increase genetic variation, besides for mutations. In prokaryotes, which is you don't know very well, they have lots of different ways to do what's called horizontal gene transfer. That's within the same generation. And basically what happens whenever a bacteria dies, it releases both its chromosomes, its single chromosome, and it also is going to release what are called plasmids. Plasmids are right down here. And when plasmids get released, the plasmids themselves are just small rings of DNA that contain codes or code for 1 or more genes, typically one to 2 genes. And so if you have a transformation, when this cell dies, its DNA is exposed, this one picks it up and gets incorporated in there. So that's horizontal, same generation. And then down here we have plasmid transfer between two. So let's say this is an antibiotic resistance. Plasmid is containing some kind of protein that causes the coat of the bacteria to become more complex. And that keeps the antibiotic from getting to the bacteria. That is going to be called conjugation. We can transfer those between. And then the third way that we can do it is because this virus picks up a piece of DNA from the bacteria. When it erupts from the bacteria and infects the other bacteria, that's called transduction. And it's going to be transferring not only its DNA, but also the DNA of its former host. And so that is a horizontal gene transfer, which happens in prokaryotes, transformation, transduction, conjugation. And far more familiar to you guys are the things that we see in eukaryotes, and that includes meiosis. And in meiosis, we have a couple of things going on. We know that during the lining up during the metaphase plate, that the chromosomes kind of line up randomly, and as a result of them lining up randomly, you can pick up chromosomes from mom or dad, but not both. And then sometimes during prophase, before they go through meiosis, sometimes there's this process of crossing over which occurs. And the crossing over is going to be exchanging genetic material between homologous chromosomes. Let's say the orange came from dad and the purple, pink, whatever that is, chartreuse, mauve, mauve, that is going to be coming from mom. So this chromosome from dad, this chromosome from mom, crossing over occurs, and now we have one normal chromosome here, and then we have one recombined, one normal over here, and one recombined. So half are normal, like this is a dad, straight from your dad. This is boom, straight from your mom. This one's a mix from mostly mom and a little bit of dad. This is mostly dad and a little bit of mom. So that is going to create some variations that we didn't have before. That's crossing over. Not necessarily a mutation, but it is a change in a DNA sequence. And of course, random fertilization. We release millions of sperm. When fertilization takes place, a few dozen of those might make it to the egg, but only one's going to make it to the egg. I'm not sure which one makes it to the egg and what their particular combination of traits is. So those are all going to increase genetic variation that we see in eukaryotes. So we can either have horizontal gene transfer or what we see in eukaryotes, which is kind of more of a vertical between generations gene transfer. Right, viruses themselves cause a lot of problems. They can cause you to get sick. We can create new viruses. You've probably heard of new forms of viruses, and they can, like we saw in the previous thing, transduction. They can take it from one to another. And in this particular example, we have the two different cycles. We have the lysogenic cycle, in which a bacteria is infected by a virus. And you can see that the virus itself gets its DNA incorporated into the host. Every time it divides, then after that, it is going to be containing viral DNA. So if you see any bacteria species that have been affected by viruses, all the succeeding organisms within that group are going to have the virus in them. So that's a lysogenic cycle, and that can be the virus can get itself transferred between generations that way. And then sometimes, more commonly, what happens is a lytic cycle, and that's where the virus infects a bacteria, takes over the machinery of the bacteria, and that causes new viruses to be formed, and then those basically burst out of the bacteria. The bacteria dies, the virus goes to infect other things. So lytic is Lyt or Lys. That's typically going to be something where you're going to get a breakdown or a killing. So the lytic cycle is going to kill the infected host, and the lysogenic cycle is a little bit more moderate. It's just going to pass it on between generations. Now, this other really cool thing happens, and I'll tell you the story about DNA, or HIV and its evolution. And what we see here is a pretty simple example. Is sometimes cells can be infected by more than one virus. And this is probably what happened with HIV, is that a primate along the somewhere in Africa got infected with one virus, and then got infected with a second virus, and those viruses ended up in the same cell, and when they replicated, they accidentally got incorporated into the same viral coat, and then they themselves started infecting other cells and created a new virus. So you can see that we've heard of avian viruses coming from birds, and we've got swine flu viruses that are coming from pigs, and then we have influenza viruses that are present in humans, and those viruses can get intermingled and create new viral combinations, some of which, like SARS, or the influenza outbreak that happened in the Spanish flu in 1919-1920, are examples of new viral combinations that result from multiple viruses infecting the same cell. So they can both create their own genetic variation, and they can introduce genetic variation into their hosts. So here's a basic example of viral replication. This is kind of the common thing. You are going to need to know this. I popped it on here for you. Just kind of looking through this, how the virus tricks the cell into taking it up. Once it takes it up, then it replicates. Once it replicates, then it uses the machinery of the cell, the protein-making machinery of the cell, to transcribe mRNA into proteins. Then the proteins themselves go assemble the new viruses, and then the replication of the DNA itself is incorporated into that, and you get the new baby viruses. This would be a basic lytic cycle. And every time you get an influenza virus, every time you get an upper respiratory infection that's viral in nature, this is exactly what's happening in your cells. So just pay attention to that. Just kind of make sure you're familiar with the process. All right, so when we go to the bodies exhibit, usually one of the things that's fascinating to me is how we get from a single fertilized embryo, like we see here, into the specialized tissue that make you now. And again, you see at the bodies exhibit. And that is basically because of chemical signals outside the cell, the makeup of the fluid that surrounds the cell, and again, our gene expression, which is going to cause cells to become moved from undifferentiated, like we see in this picture here, the proceeding of a fertilized embryo to a 16-32 cell stage, into what's called a blastula, which is, again, all undifferentiated tissue, and then as it forms the particular tissue for the particular organism. In this case right here, we have a tunicate, and you can see here the different parts of the embryo have been injected with dye, so we can see where that dye ends up, and they end up in different tissue within the organism. So chemical signals from outside, gene expression inside, differential gene expression inside, can result in unspecialized cells becoming specialized cells. And we'll go over the mechanics of this a little bit more in class. We won't spend a ton of time on how they become specialized, but we do want to understand that the chemical signals are mostly what's causing that, along with differential regulated gene expression.